Hello, good morning. So we're going to continue on our talk about our inherited traits. This is one thing that can happen, not necessarily in humans all the time, but it is does happen in plants very often. So incomplete dominance is just sometimes an immediate form of the dominant traits appears. So this means that mixing of colors or sizes occur. Incomplete dominance may occur when a dominant allele and a recessive allele are inherited. The offspring will have a mixture of these two. For example, in some species of sheep, gray fleece results from a dominant white fleece allele and a recessive black allele. Incomplete dominance also occurs in petal colors in some species of plants, just as so does the picture above. So it's pretty much saying that it's just pretty much a blending of two different colors per se from the dominant and recessive gene. Now, codominance is very different from incomplete dominance. So it shows the blending of traits, the other one does, but for this one, codominance results in both alleles being expressed at the same time. In cattle, horses, and dogs, there's a color pattern called Rowan. This color pattern appears when a dominant white hair allele and a dominant solid color allele is inherited. The offspring has hairs of each color intermixed, giving the solid color a more mudded or molted look. So pretty much just like the flower shown over here, incomplete dominance of this dominant and recessive gene is going to turn into a pink because it's blending the colors. Codominant, they're both going to appear on the organism itself. Now, there's sometimes multiple alleles. Blood type would be a good example. So every offspring inherits one allele from each parent for a total of 2 alleles. However, sometimes one trait has more than 2 alleles. So for example, there's 3 types of alleles for blood type, A, B, and O. The A and B blood types are codominant and O is recessive. You see that there's possible recombinations of alleles result in one of the 4 different blood types. Multiple alleles are not found only in blood types, but it can also be in fur color in some rabbits as a result of multiple alleles. So we see here that A and B are dominant, O is recessive, and that's going to give us those 4 different blood types, A, B, and O. And don't forget that some of them are going to be positive or negative also. Polygenic inheritance. So some traits are controlled by more than one gene. In polygenic inheritance, these different genes are expressed together to produce this trait. Human height is an example of this. If the mother is 5 feet 2 inches tall and the father is 6 feet tall, then you might think of that as the offspring would be 5 feet 7 inches. However, there can be a large variation among the height of the children. This fact is due to multiple genes working together to produce that specific trait. Remember, height, skin, and weight are all controlled by many different genes, and obviously your environment as well. What kinds of things have you learned in your life? Maybe you know how to paint, maybe you know how to ride a unicycle, or maybe you know how to solve very complicated math problems. Whatever your abilities are, they are acquired traits as the result of learned behaviors. The traits you inherited can be affected by your experience. For example, humans are born with teeth, vocal cords, and tongues, all of which can enable us to speak. The language you learn to use depends on your environment. You were not born speaking a particular language, but you were born with the capacity to learn languages, whether a spoken language or a sign language. The ability for language is our inherited trait. The language or languages you use, however, are acquired traits. The combinations of inherited traits and acquired traits help many organisms survive in their environment. The fox squirrel, for example, has inherited traits from its parent that help it survive in its environment. The squirrel also acquires traits that help it survive by learning behaviors from its parents and by interacting with its environment. So everything can be also manipulated by our environment. So as we see here on this picture, we have this couple that is very fit. So it's not like if they have a child, it's not just going to be born with a whole bunch of muscles. They have to work towards it. They have to acquire the muscle gains that the parents have also. Organisms interact with their environment on a regular basis. Some of the ways you interact with your environment may be Sweating would be getting chills, things like that. You may spend time with friends, breathe fresh air, exercise, and enjoy a sunny day. Unfortunately, some of these interactions may change the way a gene is expressed. Gene expression determines how inherited traits appear. The environment can lead to changes in gene expression in several ways. Certain chemicals in tobacco smoke or exposure to the sun's harmful ultraviolet radiation may cause changes in the way certain genes behave. These changes alter the way an organism functions and may produce different traits that would normally have been expressed. Though not a guarantee, these changes may cause cancer and other diseases. Not all changes in genes caused by environmental factors get passed on to offspring. For example, too much UV radiation can damage the DNA in skin cells to the point of causing cancer. These damaged genes, however, do not get passed on to the next generation. In order to pass on genes that were changed by the environment, the change must occur in one of the sex cells, egg or sperm, that formed that offspring. Because the genes that were changed were most likely in the body cells or cells other than sex cells, they can change genes without not being passed to you, and would instead affect all the individual with the changed genes. So it's very important for us to know that it would have to make a change in the DNA, and it has to make a change in the sex cells for it to be passed on to your offspring. So we're going to move on to talking about Mendel and all of the things that he did to be able to hypothesize that these traits are being passed down from generation to generation. So like all other organisms, the cardinals, a type of bird, pass down their traits to their offspring. To better understand heredity, the passing of traits from parents to offspring, it is important to learn about the history behind the science. So in 1800s, a European monk named Gregor Mendel studied heredity. Mendel's job at the monastery was to tend the garden. After several years of growing pea plants, he became very familiar with 7 possible traits that plants could have. Some plants grew tall, while others were short. Some produced green seeds, while others produced yellow. Mendel's studies became some of the most important in biology because he was the first one to quantify his results. He collected, recorded, and analyzed data from tens of thousands that he ran. The experiments Mendel performed involved transferring the male flower part of the pea plant to the female flower part of the desired trait. Mendel wanted to see what would happen with pea plants as he crossed different traits, short and tall, yellow seeds and green seeds, and so on. Because of the detailed work of heredity, Mendel is often referred to as the father of modern genetics. So he was cutting and putting several parts of the female and male parts of the flower onto each other just to make sure that he can get a variation of these traits to see what would happen. So he was the first person to pretty much experiment with genetics. When Mendel cross-pollinated or crossed a tall plant with a short one, all of the offspring were tall. The tall plant and short plant were called the parent plant, or the P generation. The offspring are called the F1, or the first filial generation. The term filial originated from the Latin term filius or filia, which means son and daughter, respectively. Mendel examined several traits of pea plants. Through this experimentation, he realized that certain patterns formed. When a plant with green peas was crossed with one with yellow peas, all of the F1 offspring were yellow. However, with a generation of F2, the resulting offspring were not all yellow. For every 4 offspring, 3 were yellow and 1 was green. This pattern of inheritance appeared repeatedly when Mendel tested other traits, such as the pea pod shape. Mendel concluded that while there is only one form of the trait is visible in F1, in F2, the missing trait sometimes show itself. So this is the beginning of him seeing that there are recessive genes that, even though in the first generation, so for example, we have these flowers, we have all of these purple flowers because it's the dominant gene, but then for some reason in this F2 generation, we see that white recessive gene come out again. So this is where he needs to start making more experiments to see why that is happening. So in Mendel's time, people had no knowledge of genetic material or its ability to carry the code in an organism's traits. However, Mendel was still able to formulate several ideas about heredity from his experiments. He called the information that carried the traits factors because they determined what was expressed. He also determined that for every trait, organisms receive one factor from their mother and one factor from their father. He concluded that one factor can mask the expression of another, even if both presents are at the same time. So very important. So today the term factor has been replaced with gene or allele. Alleles are the different forms of a gene. Pea plants have one gene that controls the color of the seeds. This gene may express itself in either yellow or green through a combination of yellow alleles and green alleles. When crossed, each parent donates one of its alleles for seed color to the offspring. The allele that each parent donates is random. An offspring's seed color is determined by the combination of both alleles. An organism's traits are controlled by the alleles it inherits. A dominant allele is one whose traits always show up in the organisms while the allele is present. So a recessive allele, on the other hand, is hidden whenever the dominant allele is present. If one parent donates a dominant allele and the other donates a recessive allele, only the dominant trait will be expressed. So pretty much what a dominant allele is most of the time it's going to be expressed unless two parents are going to be carrying that recessive gene. But this is mostly review for you guys, so I am not too worried. I know you guys know most of this stuff already. So the traits that we have on present that we see because of the combination of alleles. So for example, peas show two different colors. Pea color is a gene, where the combination of alleles determines how the gene will be expressed. To represent this, scientists who study patterns of inheritance called genetics use letters to represent the alleles. A dominant allele is represented with a capital letter and a recessive allele with a lowercase letter. When an organism has two of the same alleles for a trait, it is called a pure breed. This is represented as two capital G's or two lowercase g's. When the organism has one dominant allele and one recessive allele, it is called a hybrid. This would be represented as a capital G and a lowercase g. Remember that each trait is represented by two alleles, one from the mother and one from the father. Depending upon which alleles are inherited, the offspring may be pure breed or a hybrid. Mendel's work was quite revolutionary. Prior to his work, many people assumed that all traits in offsprings were a mixture of each parent's traits. Mendel's experiments where traits appeared in the F2 generation that were not in the F1 generation disproved this idea. So pretty much Mendel was the first person to, one, study genes, and two, make sense of these recessive genes that are going to be showing up in this F2 generation that did not originally was able to be seen in the F1 generation, even if one parent had that dominant allele. All right, so Punnett Squares. To determine the probability of inheriting traits, geneticists use a tool called a Punnett Square. To construct a Punnett Square, it is important to know what trait is being considered and whether the parents are pure breed or hybrid. So just know that the Punnett Square is very easy to use. Also, you just put, like, for example, these are going to be a heterozygous father. This is going to be a heterozygous mother. We are going to bring these letters down, these letters across, and thus we have our Punnett Square. Now we see here that we have two of them. They're going to be homozygous. This one's homozygous dominant. This one's homozygous recessive. But we have these two heterozygous offspring. So you're going to have this figure, but then at the same time, that would be our genotype, but then our phenotype per se would be, let's say this capital B was brown fur, this little b, let's say it would be white fur. So we could say that we have a 3 would be brown, and then 1 would be white because of that dominant gene that is over here and over here for the heterozygous offspring. So I'm actually going to pause there. We can continue the other time. Thank you.